Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Interventional Cardiovascular Device Development, Considerations in Clinical Trial Implementation. My name is Dian, and I'll be your X-Talks host for today. And today's webinar will run for approximately 60 minutes, and this presentation will include a Q&A session with our speakers. This webinar is designed to be interactive, and webinars works best when you're involved, so please feel free to submit any questions you may have and comments to our speakers throughout the presentation. And you could do so by using the questions chat box, and we will try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. And should you require any other assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode, and please note that this event will be recorded and made available for future download. At this point, I would like to thank Novella and the Cardiovascular Research Foundation who helped develop the content for this presentation. And for now, I would like to introduce our speakers for today's event. Our first speaker, Eric Diestad, who has more than 16 years experience in all facets of clinical research, including 13 years in medical device clinical research, ranging from one of the top 3 medical device companies to startup companies. He has been responsible for the development and management of more than 20 IDE studies, including protocol development through final report and submission to regulatory authorities. His experience includes primarily Class 3 devices with indications in neurology, cardiac, vascular, and spine disorders. Geographically, Mr. Diestad has significant experience in managing trials in the United States, Canada, Europe, Hong Kong, and China. Our second speaker for today is Dr. Ori Ben-Yehuda, who is a distinguished clinical cardiologist and co-author of numerous scientific publications in top-tier medical journals. Most recently, Dr. Ben-Yehuda was Vice President, Cardiovascular Clinical Research at Gilead Sciences, and prior to that, he was Professor of Clinical Medicine in the University of California in San Diego and the Director of the Coronary Care Unit at UCSD Medical Center. Dr. Ben-Yehuda also served as the Deputy Editor for the Journal of the American College of Cardiology. And without further ado, I would like to hand over the mic to our first speaker, Mr. Eric Diestad. And Eric, you could begin when you are ready.
Great, thank you, Dan, and thanks everyone for joining. I think this is a very important discussion that we're all approaching here. In that, this first slide, I'm kind of getting off track a little bit in the actual design of the clinical trials, but getting to why we need to execute these well and efficiently. So there's really 2 aspects of this. There's the fiscal responsibility of running a study effectively and correctly the first time, but also the fact that 50% of all non-communicable deaths continue to be due to cardiovascular disease. That's amounting to about 17.3 million deaths currently and 24 million deaths globally annually by the year 2030. So, whether it's for new products being innovated or basic science research, executing these clinical trials well and effectively are leading to better care, which hopefully will drive these numbers down. And the fiscal aspect of it, you look at the U.S. is currently expected to lose about $4 trillion between 2014 and 2018, 2028. That's a huge dollar figure, and that loss, about 18% of that loss is due specifically to the decrease in daily adjusted life years. And for those of you not familiar with that metric essentially measures the productive years of one's life. So as that number goes down, their productivity decreases and contribution to society decreases, which in turn drives up costs both in their decreased productivity as well as increased medical costs. So by designing these trials, getting innovation available to patients sooner, all leads to improvements in these numbers. And from a medical device standpoint, and I'm going to have a fairly heavy twist on medical device in my presentation because that is where my background is. By this year, we're expected to see about $66 billion spent with or through medical devices for the treatment of cardiovascular diseases. And the percentage of medical devices being used to treat cardiovascular disease and disease in general is increasing for a number of reasons. One being through medical devices, you're able to acutely treat a disease state and location versus systemic treatment through pharmaceutical, hoping for a localized effect. Going into that just a little bit more, we're seeing a huge increase in clinical trials globally. That makes sense on a number of fronts. One is the fact that innovation is increasing. So as there's more therapies, obviously going to lead to an increase in studies, and as well as global agencies, global regulatory agencies are all increasing their regulatory data requirements. So that, as well as increasing the number of clinical trials that are being executed. Now, interestingly, though, as the number of clinical trials and the need for these trials increase, reimbursement for many of these products are decreasing. So while the cost to both bring these to market and through post-market studies to keep them on market is increasing, the return on investment is decreasing, which, again, from a fiscal standpoint, leads to the necessity to run these studies both operationally and cost efficiently from the beginning. Talking about the cost, medical devices and really therapies in general are all in the crosshairs from a financial standpoint, both from public-private payers as well as hospital purchasing departments in themselves. Purchasing departments and reimbursement organizations are looking for pricing concessions, which is driving down the price and reimbursement for a lot of these therapies and products. Utilization pressures as well. I don't think there's really any institution that's going to turn down a therapy if they're able to see that it's both safe, efficacious, but now more so has an economic benefit as well. So in other words, part of that utilization pressure, and we'll talk about it in a few slides, is the health economic aspect of it is, in using this therapy, what is a long-term both health but also economic benefit of that versus other available therapies. And of course, we're seeing numerous reforms in healthcare. Most prevalent in the U.S. that's been discussed, obviously, is Obamacare and the effect of that. And for the medical device arena is the device tax, which is a direct tax and is affecting R&D dollars and again, driving that need for cost-efficient trials and doing this correctly the first time as we run a trial. So jumping now into the kind of the meat of the presentation, or at least of my part of it, is what are some of the key challenges to these these cardiovascular trials. So one of the main ones, and probably not news to anyone in this group, but is deciding on what the comparator is going to be. When running that trial, what are you going to compare your therapy to? Is it going to be pure standard of care? Is it going to be a specific competitive device or competitive therapy in general? And if so, how do you choose that? Geography plays a big part into that. As we see a huge increase in global trials, global trials being executed under a single protocol, that compounds the complexity of it. Going back, 10, 15 years, the model was more often run a trial in Europe, run a trial in the U.S., run a trial in PAC Asia. Now the trend tends to be trying to blend those into fewer protocols and ideally a single protocol. So across all these geographies, how do you decide on a comparator that's going to be reflective across all these different geographies, all these different healthcare systems? So some key things to consider there: relative clinical effectiveness, understanding what the treatment algorithms and paradigms are across all these geographies, and how both your product, your therapy, as well as your comparator is going to play out across all these different patient populations. Availability of other treatments for conditions. Particularly as we start to look at some less developed first world countries, some in PAC Asia come to mind, South and Central America. Do they have the availability of these treatments that you're, both of your own? Do they have the tools necessary to implement it, but also of your comparator and what you're using for standard of care, other comparative models. And also important there, as you look at the endpoints, do they have the means to consistently measure the endpoints? If you look at U.S., Europe, certain parts of PAC Asia, certainly, what they have available for such things as imaging technology is vastly different from lesser developed areas of PAC Asia, India, South America. So making sure as you develop those endpoints, it's going to be measurable across all of your sites and geographies. Quality of care, I think it's fairly self-explanatory, but do all of your sites have the general same level of quality of care? Because if a certain handful of sites have a significantly better or worse general quality of care, that's going to affect your outcomes and your endpoint measurements. Health outcomes, I'm going to hold off a little bit until a couple slides from now, but how are you going to measure the health outcome endpoints that are important for each one of these sites and geographies? Blinding is another difficult piece, particularly as it applies to medical device trials, but certainly across any trial. For medical device trials, when you're looking at an invasive procedure, an implant or use of a device, trying to blind that is exceptionally difficult. Sham procedures quite often are not considered ethical, so those quite often aren't available. And important, however you decide, if you decide to blind or not, if you decide to, how are you going to implement that and make sure you maintain that blind? And more trials are going really to the direction of not blinding. For a period of about 7 years, 2000-2007, only about 14% of cardiovascular trials were indeed blinded. So, the gold standard obviously is still prospective, randomized, blinded trial, but if you're not able to maintain that blind, then you've really defeated that purpose and damaged the trial more than helped it. And the FDA is becoming much more involved in this. At one point, going back historically, if you said you're going to blind it, they said great and kind of moved on. Now they're really digging deeper into how are you going to blind it, how are you going to maintain that blind. If a blind is broken at a particular site or with a particular patient, how are you going to handle that? And having that developed and tightened up front is going to expedite approval of that study. As an example, a trial that I ran a few years ago, the general protocol was approved by the FDA basically after one round of review, but we spent the next 4 or 6 months negotiating with them how we're going to design and maintain the blind. So keep that in mind as you think about how or if you're going to do a blinded study. And part of that also is the site and study staff, making sure they have the resources available. Even the fact of electronic medical records, EMRs, is making blinding across site personnel at a single site much more difficult because everything's entered in the EMR, they all have access to that, and whether intentionally or accidentally, it's not too difficult for them to come across some information that unblinds them to a particular patient. Health economics, I've mentioned this a couple times. Again, going back to when I was first designing trials and executing trials, health economics was, a far second, third thought as to measuring that early on in a product development life cycle. So in other words, we really didn't think about collecting any kind of health economic data, certainly that first in man or even that pilot or pivotal study where the focus was almost purely safety and effectiveness. But now with both economic inputs that we're seeing, as I mentioned, from various payers, from hospitals themselves. Collecting this information up front early in your trial is going to be critical to actually having your product adopted and used even once approved. Some of those tools, patient-reported outcomes, looking at quality of life, are all things that are going to drive some of that, what I often call quality health economics. So it's not health economics such as quality adjusted life years, but metrics that lead to institutions seeing the economic benefits. So clearly, if you do quality of life surveys, subjects are returning to work sooner, feeling healthy longer, sooner. Those lead to decreased follow-up needs and decreased cost to the institution. Inpatient assessments such as OR time, blood loss, radiology use, all of those as well lead to identifiers of decreased costs. Because if you have a decreased OR time, decreased blood loss, decreased use of some of the tertiary units within the hospital, that all equates to decreased cost for the institution and leads into the economic viability of your product at that institution. And then looking at cost effectiveness data as well, and we list quality adjusted life years here, but typically for a pivotal study, you're not going to use, for most geographies, you're not going to be required to do that level of health economics. So that typically draws out your follow-up period significantly, but certainly some of these other health economic endpoints can help tell your story of the viability of your product from an economic standpoint. CMS, I'm going to touch on this briefly because it is kind of purely a medical device aspect, but if you're going to try to get CMS reimbursement during the trial, you need to plan for that far in advance. The approvals take a significant amount of time. For those of you who have done this, you're familiar with this timeline, the complexity of it. And once you get national approval, then you need to go to each state, get approval there, understand what their submission process and approval process is. And then even once you get to that point, having the resources in-house to manage this process is extensive. At one company I was at, we had an entire department of about 45 people that did nothing but manage this process for a clinical trial. So you really need to do a cost-benefit analysis as to whether this makes sense for your product and for your study. Private insurance, you need to be careful of this, particularly as you look at specific countries in Europe, that this varies considerably in the sense of some geographies are going to say, yeah, it's a clinical trial. You're expected to provide the therapy, whether that's a device or a drug. Everything else falls under more or less standard of care. Other geographies are going to say anything listed in your protocol, whether it's for the comparator or for the test group, if it's in the protocol, it's covered by the study, and we're not going to reimburse any of that. So you need to have a good understanding of what that's going to look like across all of your sites as you kind of plan for this. And again, this is something that you need to be looking at very, very early on in the design phase because it's going to affect both your timeline and the cost of running the study. Another clinical challenge, one of the larger ones, you were probably all very familiar with the importance of selecting sites, but specific to cardiovascular studies, some of the things you need to consider: sports certified, obviously, and technical expertise. So one of the things to consider here is, as you select sites and you select investigators, do they have the expertise and the technical training to use your specific therapy? And again, this becomes more critical with devices, but do they have the experience of actually using your device or very similar like devices? Because if they don't, then you're going to have this learning curve that's going to affect outcomes and therefore the quality of your trial. Proven operator performance, kind of the same thing there. Are you able to demonstrate, or are they able to demonstrate that they have the experience required to use the therapy that you're testing? Trial-specific infrastructure, we talked about this a little bit earlier, but making sure going beyond, do they have adequate patient space? Do they have adequate site coordinator resources? Going far beyond that, but do they have the imaging technology that you required for your study? Do they have the capabilities to transfer data, whether it be patient-reported outcome data collected electronically or images or other tests? Are they able to transfer that information the way that you're intending? And as databases get more complex and more integrated into single systems, if you're intending that on that data being provided directly electronically into those data management systems, do they have the capability of doing that? Understanding what their EMR is like. Are you going to have access to that? How are you going to monitor that? Making sure you understand all of that. Understand their startup timelines, what other trials they're running, and that's not that unique for cardiovascular trials, but I think it takes a little bit more time to dig into it again as you look at specific geographies. For example, most cardiovascular trials are going to require imaging data. So you look at, say, Germany, which has a specific radiology submission that you have to do, greatly expands that startup timeline. So it doesn't mean you don't go to countries with these kind of limitations, but making sure you understand what those limitations are up front. Hopefully you have some type of evidence and experience with these sites of providing high-quality data and understanding what the metrics and how you're going to measure on an ongoing basis what that quality level of quality is going to be. Certainly monitoring is part of it. More and more use of risk-based monitoring and pulling specific metrics from the CRFs and queries, et cetera, are all leading to a more proactive measure of that quality, but having that metric for each one of the sites established up front. And the experience of clinical research coordinators, I think, it used to be more so that when you looked at research coordinators, your concern was not so much do they have the therapeutic experience, but do they have the research experience? Again, aging myself a bit, but going back 10 years, 10, 15 years, the question going into a site coordinator was, have you run studies and actually have the time to be a research coordinator? Because quite often they were, the surgeon's nurse, and now they're being asked to be a research coordinator as well. And I think that's still the case, but I think there's a second part as some of these larger institutions essentially develop almost internal CROs and have a bank of research coordinators, understanding how they're going to assign those research coordinators, and I'm making sure that they are research coordinators by training, that's their job and that's all they do, but do they also have the therapeutic experience and expertise that you're going to require from them for the trial? So kind of a summary slide here. I think kind of went through this fairly quickly. I know we have a lot of time for some questions at the end, so please don't hesitate to put those forward. But I think protocol development, Ori is going to go into this quite a bit as well as far as endpoint selection, endpoint measurement, and how to implement a lot of that. Strategic partnerships with highly efficient sites and vendors. So if you're going to outsource any of this work, make sure that whomever you're going to outsource it to has the expertise that you're looking for. So if it's a device study, do they have designated experienced device staff? Do they have staff that are specific to your therapy in disease state? So in this case, for example, we have a cardiovascular vertical, and everybody within that vertical are highly experienced and trained within that therapy or disease state and therapies. So making sure that they all match and have that experience that you're expecting of them. Targeted clinical execution driven by skilled study teams. So again, very similar to what I was just talking about, but also making sure that there's going to be a smooth integration of execution of all the levels of the study, from various vendors to the sites to the sponsor company, and if you're outsourcing to the CRO, making sure there's a clean model and clear expectation as to how all that's going to be integrated. So that is the summary of what I'm presenting. I think Ori is going to present next, and at the end we'll have plenty of time for some questions. So thank you all for joining, and as I have mentioned a couple of times, please feel free to reach out with any questions.
Thank you, Eric, for that presentation. So now I will hand over the presentation to our second speaker, Dr. Ben Yehuda. And Dr. Ben Yehuda, you may begin when you are ready.
Thank you, everyone, for your patience. And what I wanted to start with is just reminding people that the approach to drug studies and to device studies is very different. And there are very clear reasons for it. And these reasons end up really influencing the philosophical approach of the regulatory agencies. So for drugs, they fall under CDER or CDR for the Center for Evaluation and Drug Research. And with drugs, we're used to very large trials now, large outcome studies in cardiovascular diseases. And what makes drug studies somewhat more straightforward is that at the sites, there needs to be expertise, but it's not as extensive as with device studies, in which you need to train the operators in a new device on how to use it. And if any side effects occur, most typically the study drug can be stopped and the side effect would be expected to resolve. That's not always the case, of course, but as a general rule. While devices, again with some exceptions, are often implanted permanently, for example, a stent once implanted in the coronary artery cannot be removed, the number of subjects in trials are usually smaller, and because of that, the studies inherently are less powered, and that has significant implications to our design of studies. And specific interventional expertise is needed, as mentioned. Pathways also differ between the US and Europe, which until recently have been the major markets where trials have taken place. This is now undergoing, as Eric pointed out, significant change as devices are being used as well as studied throughout the world, especially in areas such as Japan, China, India. In order to get approval, you often need to actually do a local study in addition. But the traditional pathways are in the US and Europe, and the pathway to device approval varies significantly between the two. Except for 510K, which are lower risk devices and are already deemed equivalent to another marketed device, in the US most of the time we need PMA application. It's a long, rigorous process and is both costly, associated with more risk to the sponsor. And so the regulatory pathway in the US is much longer compared to the CE mark pathway in Europe, which is handled by notified bodies, which are actually private organizations which are certified to give the CE mark, and the pathway to approval is often much faster. Interestingly enough, when one looks at actual time to market, because in Europe you often need to have reimbursement, which is a long process in Europe, particularly in some geographies such as the UK, France, etc., the overall time to market may actually be similar. But still, the situation is that for many devices, they're available in Europe and are not available in the US because the pathway on the regulatory side is seen as onerous, expensive, and very long in the US. This has led to a lot of frustration, both on the part of clinicians as well as the public and Congress, with the US lagging behind in ultimately patient access to innovative products. And particularly in the device field, we're talking about innovation. So as a prototypic example, drug-eluting stents introduction were delayed by several years compared to Europe. And even now, there are much fewer, and only the big strategics have drug-eluting stents on the market in the US versus actually dozens of approved products in Europe. So there is now added concern that the US has lost its edge in innovation. In order to address this, the FDA under CDRH, the Division of Devices in Radiological Health, has put forth a new plan called the Early Feasibility Studies in the US. And the goals of the studies is to provide increased patient access to beneficial medical devices and to maintain and regain US leadership in innovation. The basis of EFS studies is small number of subjects, the device is intended for a specific indication, which may be early in development, sometimes even before the device design has been fully finalized. This means that less preclinical data is necessary, and the whole process is viewed as an iterative process in which, over time, we learn from the results of the EFS, and the product itself, the device itself, may undergo further refinement and development. It may not be the first clinical use, so there might have already been a first-in-human study, but it should be early on in the process. And in terms of getting EFS studies approved, from the FDA perspective, the emphasis is on risk assessment and risk mitigation. Still, it does enter under the IDE framework, or investigational device exemption, and so it is still a relatively robust regulatory pathway, but the key differences are that the FDA is very engaged, timelines are shorter, and the FDA is really helping companies move this process along. The key to a successful EFS program is early engagement with the FDA. It is possible to combine the EFS study together with the CE Mark study, but it is important that a significant portion of patients be recruited in the U.S. A good goal is at least 40% being from North America, and in general, the FDA has accepted Canadian data together with U.S. specific data as a general rule. Clear risk assessment needs to be defined early on in the process, and while limited device testing may be acceptable, the tests themselves still need to be done correctly according to regulations, etc. Now, if we start with an EFS program, obviously that will not lead to product approval in the U.S. It's the first stage towards an ultimate pivotal trial that would lead to PMA approval. And so it's extremely important to have a long-term plan. And the long-term plans that succeed are based on, one, a real medical need, a good trial design in which the endpoints are clinically relevant, are achievable and measurable. Studies need to be adequately powered, but for us as we implement studies, most importantly, they have to be feasible in terms of ability to enroll, ability to collect good data, and actually have the patients be in the correct follow-up in order to have robust data when it comes to the day of panel in front of the FDA. Regulatory pathways have to be defined, US versus Europe, and these days we're seeing earlier entry again through EFS and other pathways into the US market, so the two have to be harmonized. Implementation is extremely important, so having the professional team that actually does its work efficiently is extremely important, but ultimately it's really the quality of the data and the quality of the submission that will carry the day. One of the things that can help immensely is having a knowledgeable and involved scientific advisory board or a medical advisory board. You need to have the right mix of highly experienced people, but it's always good to also have people who are ready to roll up their sleeves and be committed to the program. SABs that meet once a year and there is no ongoing involvement with the product development are unlikely to really be effective. There has to be a vision for the entire program, and most importantly, robust project management. There are a million moving parts in a good clinical trial, and in order to have things work on time without delays, it's the most complex orchestra that needs to be conducted, and it's really the project managers who are the conductors in this system. I already mentioned the differences between devices and drugs. Drugs, yes, we need to read the investigator brochure, we need to understand side effects, but most of the time the administration is relatively straightforward. For devices, we really need to have adequate training of physicians, and that may involve doing animal lab training in order to be able to be ready to use it in humans. We also would consider doing run-in phases of the study with the device, so that the actual data that will be presented is after some training has occurred with the device. And this obviously is a challenging area of making sure that with novel devices, we strike the right balance between bringing innovation early to patients and physicians and interventionalists being fully trained. And obviously, as already mentioned, defining the endpoints very carefully. Time and again, we see protocols that were not done with full consultation, and the result is that the endpoints are fuzzy, and then they don't meet regulatory muster and make the life of, for example, clinical endpoint committees very difficult, and most importantly, don't really capture the true data in the trial, and that will raise significant issues upon review. So what are some quality issues that we should focus on? Well, one is the protocol should be clinically relevant, as mentioned, should be enrollable, but it also has to be written very clearly. A good practice is to actually have a team go and put together scenarios of patient enrollments and different pathways. What happens on day 3 if the patient can't come in? What happens if a lab test is missing? How do we address certain contingencies? A clear protocol is the best way to avoid protocol deviations, which create enormous amount of work and can detract from the quality of the submission. And finally, the best protocols are simple protocols. The more complex they are, the more protocol deviations there will be, and ultimately the very complex protocols tend not to give clear clinical answers at the end. Site selection is, of course, very important. There's always the tendency to select sites that have been the best enrollers in other trials. Be careful of the extremely high enrollers. Invariably, there may be some quality issues, and you have to be very careful that a high enrolling site has the resources and bandwidth to do high quality work. A very good test, and one of the issues with high enrollers is that it tends to mean less PI involvement. A site that does not have robust PI involvement will make mistakes, will be open to audit findings, particularly in an FDA audit. There will always be situations where a signature is missing or a date is not written correctly. Those could generate 483, but typically would not if the FDA sees that PI involvement and care of the patient is present. So this is a key issue in choosing sites. Related to that is selection of countries, and one has to be careful to understand the landscape for clinical trials in different geographies, and what are the incentives for enrollment and how it may lead to inappropriate enrollment. And there have been cases in various geographies in which the number of enrolled subjects has been disproportionate to the prevalence of the disease, for example, raising questions that in extreme cases, issues of fraud, etc. So be careful to align the incentive with the local practices and making sure that speed does not compromise quality. Now, in ultimately having the best data, particularly in device studies, we often need core labs. Core labs are extremely important in multi-center trials because they allow a standardization of the measurements of the results across sites. Simple exercise such as quantitative coronary angiography would give very different data at a local site compared to a standardized approach done by highly skilled, scientifically rigorous technicians with the proper clinical oversight. In addition, when submitting data to the FDA, data that is done from core labs that are not proficient and are not professional will not be acceptable. You need an audit trail. The data has to be reproducible and attributable. And so beware of analyses that are done by fellows, offshore, and various other combinations that may be cheaper at first blush but do not pass regulatory muster or scientific muster for that matter. CECs, or clinical endpoint committees, have become standard in most studies and are a very important part, again, in order to make sure that there is uniformity across sites in the multi-center studies. Studies are ultimately about endpoints, and even a small a number of endpoints that are incorrect or have been missed, not collected, can sway a study from being positive to negative or giving incorrect information. So we want to make sure that there's adequate collection of endpoints, that we haven't missed endpoints, that we really canvassed both the site records as well as patients with specific questionnaires. For example, have you been hospitalized for any chest pain related issues over the past 6 months, and this could be at another hospital, is an important question because the patient may not actually remember whether they had an intervention or not, and then you need to obtain the source documents in order to see if an event has been missed or not. We want to make sure that the dossiers are adequately prepared, identified all events, and that the material is organized well. Medical charts tend to be very voluminous these days. You could be looking for a needle in a haystack, and you need professional organization to go through the chart and make sure that no relevant data is missed in the CEC evaluation. In order to have good data management, we need clear case report forms. We need to continuously review the data as it comes in, identify outliers, and have targeted audits in order to make sure that the data is filled correctly. You need clinically knowledgeable people to identify these errors so that they can see that in this particular clinical situation, this lab value or that lab value is not reasonable and needs to be queried. Ultimately, the study results will be presented in TFLs, or tables, figures, and listings, and thorough preparation of the table shells is absolutely critical. It's a good idea to run these with blinded data. It allows for identification of errors to be picked up prior to database lock and unblinding if the study is blinded. These reviews should not be rushed because once everything is locked, it is too late to change the primary data set. So I'm going to turn it over to questions, but before doing so, I want to leave you with a quote that I think is applicable to what I've been discussing. The longest route to device approval is through a shortcut. Time is of the essence, but shortcuts often lead to greater problems and delays down the road. So with that in mind, I'm happy to answer any questions, and I'm sure so is Eric. Thanks again for your attention.
Thank you, Dr. Ben Yehuda, for that presentation. And now I would like to invite our audience to keep sending in your questions to us using the chat box at the bottom of your screen. And for now, we will start with a couple of questions we have here. And the floor is open to you both, Eric and Dr. Ben Yehuda. And the first question I have is, what is the biggest challenge when conducting a CVT device trial?
Eric, do you want to take that one first, and then I'll add?
Yeah, I think we touched on a lot of the key things, but I think Ori did a great job outlining the critical piece of selecting the correct endpoints. I think we've seen trials in the past that either selected the incorrect endpoints and/or selected endpoints that could not be consistently and accurately measured. And I think we've certainly, at the end of some trials, ran analyses and suddenly realized what we want to be measuring, we can't really measure. We're not getting consistent measurements across all sites. So I think that's one part of it. And I think site selection, not only from an enrollment perspective, because I think that applies to any trial and any therapy, but from a cardiovascular study, as Ori and I both talked about, is making sure that the users are indeed properly trained. Ori mentioned doing cadaver labs, animal labs, in doing some training and enrolling patients also help from a data quality perspective.
I would add to this that a clinical trial really has a lot of moving pieces, and at the end of the day, even one area that falters can affect the entire trial. So it's not as if each one of these elements, if they're good, will carry the day. If you fall on regulatory compliance, the trial will fail. If you fail on endpoint selection, it will fail. So each one of these elements ultimately needs to have the proper attention. I will say that a key issue over the past few years has been cost. and we're really trying to find innovative ways in order to afford clinical trials and reduce the cost. And some things that have come along in the past few years have been doing trials within registers, or registries as others call them, so that or within the electronic medical record. But we'll definitely need to move towards more innovative platforms of data collection as well as enrollment in order to reduce costs, which are a major obstacle.
Great, thank you for that. And our second question is, how do you go about choosing an endpoint?
Maybe I'll tackle that one first. I think there are basically two overriding considerations. One is clinical relevance, and the other is regulatory relevance. So while we may believe that an endpoint is clinically relevant, the FDA may not yet have accepted it based on their review of literature and historical precedents. But the first is really to start, does the endpoint make sense from a clinical perspective? Does it affect the two things? Does it measure two aspects that ultimately are critical? One, does the endpoint reflect the savings of lives? And the other is, does it affect the quality of life? Because ultimately for patients and regulators as well, there really are only two important things. Will it make the patient live longer? And two, reduce their symptoms, or another way of saying it, improve their quality of life. And so we really have to keep those issues in mind when we choose endpoints for a study.
Great, thank you, Dr. Ben Yehuda. And our next question is coming in from an audience member, and the question is, have either of you attended an FDA review panel meeting, and was the drug or device approved?
Eric, do you want to start with that one?
Yeah, sure. Yeah, I've attended probably 5 or 6 panel meetings that were specific to devices that I was, or studies that I was managing and taking part in, and then some additional panel meetings simply from an audience standpoint. And all but one of those did eventually get approved through that panel meeting. So yeah, there's certainly, I think, part of being successful at the panel meeting clearly is how successful is the study, but also making sure you have the right people at the table and knowing hopefully ahead of time what the panel's main concerns or questions are going to be. So for example, a couple of those, their main questions and concerns had to do with the statistical analysis and the methodology. So making sure you have the right people at the table that can walk them through what that methodology is, particularly as we look at some of the new statistical models that we're implementing. So I think that's kind of what I found one of the keys to being successful at that panel meeting.
And I don't have much to add to that except to say that there is always some element of preparedness, some element of the quality of the study itself. A lot of work really needs to be done ahead of time in terms of work with the FDA on what were the requirements for approval of the specific device. And in addition, there is, as always in life, a little bit of issue of who are the panel members. And we've seen some situations in which both devices and drugs have come back, have initially been denied, and have had follow-up panels, and either more studies needed to be done or more analysis of the data needed to be done, and that has delayed regulatory approval. But one of the things that can help protect from that situation is to have as much agreement with FDA ahead of time on what would be necessary to gain approval for the device.
Great, thank you for that. And just a reminder to our audience members, should you have any questions, do submit it via the chat box in your control panel. And our third question would be, when is it appropriate to use a sham procedure?
Yeah, Ori, do you want to start with that one?
Yeah. So it would be appropriate to use a sham procedure if it does not in itself confer added risk. So, for example, many years ago there was a study of a device for angina which involved doing counterpulsation with an external device. And this was part of a study called the MUST-ECP study. And they actually administered treatments, but just at a much lower pressure. So there was that sham procedure had the potential to perhaps have some, but more limited efficacy. And the study was still powered and indeed was a successful study and led to approval, that they were able to show a difference between full treatment and what was called the sham treatment. But the basis for the sham treatment is that there would be no actual or very limited risk to the patient. So that would be a situation in which a sham procedure would be possible. There are situations in which doing a sham procedure is just not possible because we won't do surgery on someone and not implant a device. Where a variant of not doing a sham procedure but actually ensuring blinding is more appropriate when we do a device versus device. So for example, in looking at newer stents versus older stents and looking at a more subjective endpoint such as angina, it's very important to blind the patient to the treatment. And there we can be innovative in our study design by, for example, putting headphones on the patient, putting a blindfold on the patient during the procedure itself, or rather an eye mask. I wouldn't call it a blindfold. An eye mask would be a more politically correct term. And then really ensuring that the people who are doing the follow-up are not aware of the study allocation, of the device allocation, so that they are blinded and the assessments of the patient, particularly when we're looking at softer, more subjective endpoints such as symptoms, such as chest pain, that would be important. So there are basically two levels, but obviously the more invasive the device is, the less we're able to do fully sham procedures.
Yeah, I think the only thing I would add to that, I agree completely. I think it really comes down to is what is the added risk of doing the sham. And I think most of the sham-like studies that I have run are involved with programmable devices. So in other words, maybe it's a PACE or an ICD that you can implant the device on all subjects, but the device isn't necessarily going to be turned on all subjects initially. Certainly,
right.
You could, to offset that risk, it comes at the end where all subjects will have the availability to have the device turned on fully. But I think that's kind of a similar model to what the example Ori gave as well. But otherwise, you try to do a surgery and not actually do anything. There's obviously too much risk with a surgical procedure to not provide any benefit.
Right. So the key to what the example you just gave, which was an excellent example, is the ability afterwards to cross over into the treatment by simply turning on the pacemaker, the defibrillator, etc.
Great, thank you for that. And our next question is from an audience member. And the question is, what should be the sample size for phase 3 trials in a device trial?
So maybe I could tackle that with a little tongue-in-cheek. Answer would be the right size, the right sample size would be such that would still be powered to measure the primary endpoint. So that on its face is the simplest answer, but there's another facet to it, which is that we also need some adequate experience with the device. So if an endpoint would allow us to, for example, only test the device in 10 patients and that would be powered and statistically significant, that would not be sufficient. So we need a certain reasonable number of patients. That will vary based on the patient population, the prevalence of the disease, the ability to find such patients. So if it's a rare disease or an infrequent disease, you'll need that whole safety issue of exposure to the device in a large number of patients becomes much not feasible and not required. So it really does vary. But for example, typically a new stent in the US would require, in order to get adequate sample size, about 1,500 to 2,500 subjects in order to enroll in a one-to-one and have adequate power and adequate exposure and experience prior to approval. In contrast, in Europe, a similar CE mark study might only require, with a secondary surrogate endpoint, 200 to 300 subjects, and one could get CE mark in Europe. So it varies by geography, varies by the endpoint, varies by the disease condition, and ultimately the negotiations and agreement with the regulatory agencies.
Great. Thank you very much for that. And well, we thank you very much for those answers. And we have reached the end of our Q&A session. And if you have any further questions, you could direct them to the email address showing on your screen. And we thank you everyone for participating in today's conference. And you will receive a follow-up email with access to the recorded archive for this event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us improve our webinars. Now, please thank us in joining our speakers, Eric Distad and Dr. Ben Yehuda. And we hope that you found this conference informative. Have a great day, everyone.